Thanks, Srini. It's a perfect segue from the discussion we just had. We'll try to explain some of the first principles that might get us to a better therapeutic option for the BTK segment. So we've talked a lot about drug-coated balloons. The predominant mode, has been paclitaxel-based solutions over the last decade. And there are a host of reasons why that is, but there's many reasons to think that paclitaxel is not the best option as an anti-proliferative drug. I show this slide all the time because I think it's illustrative of the important first principles, and that is that in order to inhibit intimal hyperplasia when you give the drug from the lumen, you just need to have enough drug concentration for enough time to get an effect. And there's different ways to deliver the drug. So Zilver would be a drug-coated stent, Eluvia would be a drug-eluting stent, or Esprit for that matter. A drug-eluting balloon, we don't have any really on the market at present, and almost all of the technologies are drug-coated balloons, which have a excipient and a drug sort of sprayed onto the balloon that leaves a coating of paint on the vessel wall that includes the drug. When you give it via a drug-coated balloon, though, you're giving a ton of drug. The payload is very high, but that drug rapidly decays away from the artery over a relatively short time. The advantage of a stent, of course, is that it sticks around for longer, and you don't need to give such a big bolus of drug. The area under the curve is what will dictate the clinical effect. So, as I was saying just a moment ago, not all drug-coated balloons are created equal. For example, this is an experimental model at our CRF labs here in New York, and Juan Granada and the team showed that if you take all paclitaxel-coated balloons, Lutonix, Impact, and Ranger, and you put them in animal arteries, in this case, a swine model, the Lutonix drug delivery actually at time 0 is the greatest, but it rapidly falls away over time, whereas the Ranger and Impact have almost identical overlapping pharmacokinetic curves. And the clinical and biological effect in terms of reduction of intimal hyperplasia and the amount of clot formation and so on is really related to the amount of drug that's there over time. So one might argue that this would predict clinical performance, and I think that's probably important. The other thing we've obviously talked about many times is that we can't get as much sirolimus to bind to the artery than paclitaxel. This experiment is simple. You take an artery, you dunk it in a solution of drug, and you replace the solution over time to give it different presentation kinetics. In every case, paclitaxel binds more avidly to the artery than sirolimus does. And then this is the last sort of data slide I'll show, and that is that if you were to look in the coronary analogy, there is a curvilinear relationship between late loss, that is how much is the thickness of the intimal hyperplasia, and binary restenosis, and this has been defined over the first decade of this century. Now, if you were to take, for example, in the human analogy, a Xience stent, which is an everolimus-eluting stent, the late loss is roughly 0.18 millimeters. If you took the same stent and put it in the same patient in their tibial artery, as was done in the Destiny study, the late loss is 4 times that. That is that the amount of intimal thickness is 4 times greater in the leg than it is in the coronary for basically the same patient population. So why is that? It's because the biology is different, and we probably need to tune our pharmacokinetics differently for the legs than we do for the heart. Okay, paclitaxel we know is not dangerous. We know it doesn't cause mortality, but there are some safety issues that people are worried about, predominantly no reflow and late embolization. And so if we want to get to the coronary paradigm where sirolimus-eluting technologies are superior in efficacy, we need to figure out how to better deliver sirolimus to the infra popliteal circulation. And you've already seen from Brian that there are drug-eluting stents that are used off-label in this segment have, in meta-analyses, shown to be superior to all other technologies, except the lesions are relatively short compared to drug-coated balloon studies. So we've tried paclitaxel-eluting stents. We've seen very little success in the Saval trial with a paclitaxel-eluting self-expanding DES versus angioplasty. Brian already showed you what we think is an important unlocking of some of these data with the Esprit BTK scaffold in the Life BTK trial. And he's already shown you that Riva and R3 have bioresorbable scaffolds hot on the heels of Esprit. There's a few other ways to get sirolimus, though, into the artery. There's an interesting technology from this company, Reflow Medical, that has a self-expanding scaffold that's really a fenestration device to allow for drug delivery. And in outside the United States studies, in this case led by Thomas Zeller in Europe, the use of this spur technology coupled with a paclitaxel drug-coated balloon shows very impressive results in terms of avoiding major amputation and freedom from clinically driven TLR. Obviously, not surprisingly, they also want to couple it with sirolimus, and here's a sirolimus-coated version that's currently in clinical trials. And this is something that might be a way to enhance sirolimus drug delivery, and the deeper RCT is an ongoing study now around the world. What about drug-coated balloons? You're going to hear a little bit more from Eric in a moment about the Magic Touch family of studies, but the other ways that you can get drug in a little bit more effectively, especially sirolimus, is to encapsulate it with a phospholipid bilayer that allows for it to diffuse and reside in the blood vessel wall for a duration much longer than it would otherwise. And so this so-called nanolute technology allows for really good tissue concentrations in animal models and a very smooth surface when spray coated onto a balloon. And as we've talked about here in this meeting before, there is a BTK IDE trial that several of us are participating in that's about to start enrolling patients in the next month. And this is a 1-to-1 RCT that's going to look at 368 patients with primary endpoints at 6 and then powered secondaries at 12 months to look at patency in a manner similar to LIFE-BTK. The Concept Medical Group has a large number of trials being done outside the US, and you'll hear from Eric about the Ecstasy and Beyond Ecstasy and HOPE registries in a moment. The other competing technology that's the dominant player is that from MedAlliance, or now Cordis, the Solution family of balloons. They have a similar encapsulated drug delivery platform that allows sirolimus to bind and adhere to the vessel. They too have a very large clinical trial program, and they have a BTK program ongoing in the United States that I understand is currently on pause as they make more balloons. But they are looking in a different trial design using a hierarchical endpoint with a win ratio strategy to see if this will be superior to angioplasty. And we'll see in the next year what these data look like. Outside the US, we've seen from Ramon Varco in Australia, the Surmodics sirolimus-coated balloon as well in the below-knee segment. In a preclinical first-in-man trial, they demonstrated excellent luminal gain. And again, I would emphasize the late lumen loss is fairly stereotypic, and that is it's about 1 millimeter for a DCB compared to perhaps lower in what you see in coronary trials. So to summarize, paclitaxel devices are safe and effective. Sirolimus and its analogs have superior efficacy in coronaries, but yet to show that same superiority in peripheral arterial circulation, both above and below the knee. These newly formulated sirolimus devices are being developed specifically and expressly for peripheral applications, and they're now actively being studied against a variety of comparators, head-to-head with PTX balloons, head-to-head with angioplasty, and so on. And the best trials in the coming years are going to show head-to-head safety and efficacy against these PTX comparators or the best-in-class standard of care. So thanks very much.